Basically, cell biology is one of the new areas in experimental biological science. It was born about 60 years ago. It came from other areas. Today, I'm going to talk about the origins of cell biology at the Rockefeller Institute. I'm going to talk about the three fundadores who were in the same laboratory, how the Rockefeller Institute was a special place that permitted the crystallization of formation of cell biology as we know it today. Obviously, it had roots in 200 or 300 years of science before. So I'll talk about three people, Claude, Porter, and Palade, their groups, their students, their postdocs, and the story of pancreatic secretion, which was the first success of cell biology. I was there when it happened. I was a student of George Palade, who is considered the father of modern cell biology. Other people did work. There were other people. This did not happen in a vacuum. But these people that I'm going to tell you about contributed most of the new ideas at the beginning. Frankly, I didn't appreciate how significant it was when I was a student. I thought all of this was normal. So today's talk is to show primarily students, and most of you are students and postdocs, Alini, how things came together. We have the impression that anything that's more than 5 or 10 years old was made of stone, like the 10 Commandments, and is very, very old. I'll show you how these things started and where some of the ideas that you use came from. Some ideas to take home. Biology is primarily experimental, unlike physics, which is theoretical and then tested experimentally. Most of biology is experimental. We don't have a big core of theory. But when something like Watson and Crick published their ideas of the duplication of RNA, DNA, this became a very important theoretical consideration. In science, we have a balance between new ideas and new methods. People usually emphasize one or the other. And you're going to have to decide what your approach will be. Obviously, you need both. I favor the methods. So you're going to hear about this with methods being one of the themes, asking new questions with new methods. Finally, all of us face the problem of deciding when it's time to publish. Should we publish a bit or the whole story? And we live in an environment which is very statistics oriented. Everybody is in a rush to publish a lot of papers. One of the things you'll see here is that by the time these pioneers published, they had a very good idea of the whole picture. This is sort of a privilege if your BOLSA and your department chiefs, your laboratories in sumos depend on grants. This is very unfortunate, but this is something all of us have to think about all the time. That'll be another theme, that by the things I show you, we're mature. Not that I skipped lots of partial papers. There weren't any. There was a small amount of papers, relative, not more than 20 papers I will discuss today. So there were mature papers published when people were ready. This was another world. Unfortunately, it doesn't exist anymore. First, I want to describe the Rockefeller Institute as the incubator. It was founded by John D. Rockefeller, who was a robber baron and made a lot of money, a fortune, in the oil business. There are 3 Rockefellers in New York. There's Rockefeller Center, which are the buildings and has nothing to do with the family. There is the Rockefeller Foundation, which essentially is a granting agency and gives away money in public health. It supported this medical school after World War II, and also participate in international health vaccination programs. And the 3rd Rockefeller is a research institute founded in 1901, and all of its money came from its endowment. Endowment is a big pile of money invested that had profits, and Rockefeller, until '54, did not have to have grants. They may have gotten a special $1 million from the family for a big new project, but this is when very little research was funded by grants in the United States. That was a post-war phenomenon. It was the first research hospital and institute. The idea that we do research in the Hospital das Clínicas was started here, a hospital dedicated to research, and people didn't pay. So we have part of this tradition here, except that SUS pays. It was organized as independent laboratories, not departments. You will see that I don't like departments. They increase the entropy. And every laboratory had a boss. And that was it. He answered to the director for his budget. They also founded and edited the Journal of Experimental Medicine at the turn of the century. And this is still one of our best journals. I'm sorry. Here we go. A man named Herbert Gasser was director from 1935 to 1953. As I said, he had absolute power. He also won the Nobel Prize for the description of action potentials. He was a neurophysiologist. Our heroes are Albert Claude, Keith Porter, and George Palade. They were all together at Rockefeller in 1946 when their chief retired. Gasser could have fired them or hired them temporarily to give them a chance to reach the level of a laboratory director, which was pretty hot stuff. And in the Rockefeller Institute, they were called Gasser's orphans because he adopted and protected them. So it was a lovely place if you were Gasser and a laboratory director, and the other people participated. Later you'll see the electron microscope was introduced during the 30s, and they had access to a microscope in 44, which was part of them being at Rockefeller. A new chapter started at Rockefeller with Detlef Bronk, who became director in 54. Remember, it was funded in 1901. So 1951 was an important date for the Rockefeller Institute. And they essentially said, here we are, where do we want to be in the future? These are the trustees, not the workers. And they decided, the trustees decided that Rockefeller would have to start a graduate student program, and that students would give the energy and vitality to the Institute. This is a pretty good decision. So Rockefeller started in 55, their first graduate students, and there were 10 of them. Now there are probably 100. And it became a university in 58, changed the name. So you may hear institute or university. Now living there was an experience. They had a doctor's dining room where people were served by little ladies in uniforms a commercial plate lunch. You went there, sat down, and they gave you the lunch. No choice. So, dessert, you either ate or not, and they served coffee. The tables were small, and you could sit anywhere you want. This had an enormous effect for the senior people, the staff members, because it provided access and the chance to have lunch with all sorts of people of the Rockefeller. And if you wanted to, you call before and say, let's have lunch together, or you go there and you find an empty seat next to somebody you never knew. You could sit down and introduce yourself. So it was sort of stodgy, elitist, but fluid. After lunch, and that was a special dining room. And when European visitors came, they came to the dining room. Technicians, other people couldn't even think about going there. This was the doctor's dining room. And the doctor's dining room was opened to students, which was revolutionary. After lunch, most people went down one floor. You walked down, you didn't walk up. You walked down while you were digesting to the library, which had a big table of new journals, the journals that came in that week. This was a fantastic open stack library where you could also meet people and talk and digest and go back to work in the afternoon. So that was the environment. Now, cell biology. Claude, who was the father of the group, in '46, wrote a paper which is the beginning of cell biology, and he pointed out that Warburg apparently was the first to attempt to segregate cell components, small particles, by means of centrifugation and relate the biochemical activity of tissue extract to preformed elements of the cytoplasm. Elegant. We can take this and make a functional description, which is cell biology is the experimental study of the relationship between organelles, between the structure of organelles, intracellular organelles, and their biological activity. This is a good beginning description of what cell biology was at that time. So this is more than cytology, more than histology, because it's not only looking, it's wanting to understand function. And in fact, cell biology is closer to biochemistry than most other disciplines. Electron microscopy, which was developed in Germany in the 30s, in Europe. Transmission electron microscopy used the wave properties of matter for illumination and increased the theoretical magnification 300 times and resolution 200 times compared to light microscopy. This was a big event. In fact, it came from physics, the wave properties of matter at high velocity, and the guy who formulated the idea, de Broglie, won the Nobel Prize in '29 for his 1924 thesis. He published his thesis in '24. Somebody tested the theory, and he won the Nobel Prize in '29. It was first developed by the Germans, Siemens, in the '30s. And they were physicists, so they just looked at surface. They really weren't interested in the microscope. They were interested in using the wave properties of electrons. But Siemens Research Laboratory took the idea from the German physicists during World War II, and it's important that they had the Palade group, Claude, Porter, and later Palade, had access to it. This is 11 orders of magnitude of magnification, and you can see the eye goes from little people to a hair, and the light microscope starts a little better and goes until a bacteria. Electron microscope starts roughly at a red blood cell and can theoretically go to atoms, but we can't really arrive there. So this is 11 orders of magnitude that more than the small child that the electron microscope can open. And this really was a new world. The Rockefeller group, as I said, was Albert Claude, Keith Porter, and George Palade. They got together in '46 when Palade, who has been in, who's an MD from Romania, came to the Rockefeller Institute. Claude had been at Rockefeller since 1929. He had been there for 17 years, and Keith Porter, who was from Canada, had been there 7 years. Now, Claude was a biologist, pathologist. Porter was a developmental biologist, and Palade was a professor of anatomy. He got caught by the war. He, in 32, he was 20. From 32 to the end of the 30s, he did medicine, and then got into the army, and then became an anatomy professor. But Palade always had a flirt with biochemistry. He always was interested in it. So these people really were at the prime of their physiological scientific career when they all got together, with Claude being a very interesting man. He was born at the turn of the century. He was about 20 when World War I started. No, 18 when World War I started. And he was in Belgian intelligence. So he didn't go to college. Didn't have an formal education. He was in intelligence during the war. But the Belgian, at that time, you had to have graduation to go to medical school. But Belgian king or somebody passed a law that said anybody who served in World War I didn't have to have graduation to go to the university. If they survived, fine. If they didn't, it didn't matter. So Claude, from 1918 until probably 1926 or 27, did medicine, which in Europe required a one-year thesis, and finished probably in 27. In 29, he went to Rockefeller. So he didn't have an European academic life. Palade did a thesis on restructuration of the porpoise kidney, three-dimensional structure. And as I said, he had enjoyed biochemistry. Porter was an embryologist from Nova Scotia who went to Harvard Medical School, no, Harvard University and Harvard Graduate School. and he was a PhD in developmental biology, who shortly after finishing school went to Rockefeller. So there they were, Claude 48, Porter 36, and Palade 36. And they were all working in the same laboratory when their chief retired, and they all became orphans, with Claude being the chief orphan. Okay? And the rest of the group continued under some other people. This was the group, by the way, that showed that genes were DNA. This was the group of Avery, McLeod, and McCarthy. And McCarthy was the chief. Claude's first paper for us was 46, published in the Journal of Experimental Medicine, and the title was The Fraction of Liver by Differential Centrifugation. And these are the papers, and he was at Rockefeller until 49. He went back to Belgium to help rebuild Belgian University after the disaster of World War II. These are his early contributions. In the 30s, they were looking at tumors, isolating viruses, and he isolated the Rous sarcoma, which was the first viral cancer. He also described small particles, which were phospholipid RNA protein complexes, and later named them microsomes. What you're going to see in this series of slides is the vocabulary of cell biology. He worked on mitochondria, which were called small particles, and microsomes, and was interested in the distribution of nucleic acids in leukemic cells. 45 was the first paper with electron microscopy, and he did it on isolated mitochondria, not cells, because they had trouble with the cells. He also wrote a paper with Porter on tissue culture of cells. They couldn't cut sections. Cells were too thick. It was to be put like putting picanha on a microscope. So they worked with tissue culture cells, which had parts that were very thin. And that's why they looked at tissue culture cells before looking at sections. In 47, Claude gave a Harvey lecture. Now, the Harvey lecture is a very famous lecture in the United States. It's in New York. It's the Harvey Society. And usually, I don't know what the number is, probably 50% of the people who give Harvey lectures win the Nobel Prize. Look at the title, Studies on Cells, Morphology, Chemical Constitution, and Distribution of Biological Function. Cell biology, but they didn't call it cell biology. Porter came in 39, was 36 when they started. He left in 61 to go back to Harvard, and then he went to Denver. And his main contributions were, as I said, EM of fibroblasts, identification of carcinoma virus, and he and Palade worked very hard on methodology for using the microscope. As I said, you can't see much from picanha if you're looking at it through a light microscope or an electron. He and Palade described the structure of the endoplasmic reticulum, and here's an important one. Porter invented a new microscope. It wasn't brand new, it was a functional microscope that made sections small enough, thin enough to be able to use the microscope, electron microscope. Porter, okay. This is my hero, George Palade. His first paper was on mitochondria. You're going to see the combination of electron microscopy and cell fractionation. This is what crystallized. And Palade stayed at Rockefeller until the year before he won the Nobel Prize. Then he went to Yale, started cell biology there, and then he went to California and worked in the medical school there in a very important medical center. He died at 94 or 95, and he was active until the end, or almost. His contributions were fundamental, like if you want to disrupt a cell, 0.88 molar sucrose was good for mitochondria then. We've learned that different molarity of sucrose is good for different organelles. Isotonic saline was a disaster. Well, you have to fix the cell and you have to stay in the cell. They had used osmium tetroxide because it was a heavy metal, or near a heavy metal, and he made it work by buffering it because osmium tetroxide is very acid. So sucrose for disruption, osmium tetroxide for fixation. He saw the endoplasmic reticulum and described it first with Porter, and then he did the fine structure of mitochondria, which was 6 years after he first isolated them. There were big pauses. He initiated his pancreatic secretory cycle, showed that microsomes are fragments of the endoplasmic reticulum, caused artifacts caused spontaneously, and finally characterized ribosomes. There was somebody at Rockefeller who didn't like Palade and didn't believe that they existed. He actually wanted to find a way to embarrass Palade. He was a big man too, but small this way. So he insisted on calling ribosomes Palade particles, hoping that when they were found to be artifacts, Palade would be discredited. These are the games people played. So this list of things really is the vocabulary of cell biology, and they were all described by this group at Rockefeller. Palade started the secretory cycle, and Porter got interested in plant cells. They were even more difficult than mammalian cells because they had plant wall, and it was hard to fix. So Porter moved to plants, Palade moved to pancreas, and Claude moved to Belgium. So the first significant contribution of the Rockefeller group was, how do you do electron microscopy? Now, there are other people trying to do it, but the main form of the early electron microscope were the recipes and the things of this group. And they readily shared this information. New York City was the point of entry to the United States. Anybody coming from Europe went to New York City. It was like Rio was 40 years ago. If you came to Brazil, you had to go to Rio. And then you could go to Sao Paulo and then to Ribeirão Preto. So there are lots of people, people doing electron microscopy who came through, and they were completely open. Now there's problems. You could fix and you could see, but the problem of continuity with the light microscope was very delicate. How do you name a structure that you see in great detail that was partially seen in light microscopy? And there was a danger you could have two sets of vocabularies, one for electron microscopy, one for light. But these guys were well trained, they were mature scientists, and they were scholars. The introduction of their now classic papers always started with light microscopy, and the connections were made. In fact, they tried to keep the light microscope vocabulary. This showed a level of maturity which would be hard to replicate today. You needed scientific journals. Why? Because if you get into an area that is new, the papers have to document what was done. So the editors have to be very demanding. The editors had to demand that the methodology is clearly written and complete. They had to demand that the quality of the tissue, the quality of the image was reproducible and good, or else you'd have a literature full of artifacts badly reproduced. So all of this information, the editors are the gatekeepers of the development of the science. And Porter, Palade founded new journals. First, the experimental medicine continues until today. The Journal of Biochemical Biophysical Cytology was the invention of Porter, Palade, and others, and it stopped in '61, and they invented the Journal of Cell Biology. You noticed biochemical and biophysical. These were key words in science in the 50s. Partly the flirt with biochemistry, and biophysical because there was crystallography became very popular. But the journal Cell Biology, which was started in '62 by the new Society of Cell Biology, published and still publishes the Journal of Cell Biology. The American Society was founded at Rockefeller on Porter's desk by Porter, Palade, and others. First Congress in 1960 had 800 participants and 240 posters. The number increased to more than 10,000 members by 2000, 40 years, and 25% of the members were living outside of the US. So it was truly international, and the result of them being open with information. Now here's something that blew my mind. In the 59 years since the founding of the Society, 32 members of the Society have won the Nobel Prize. That's one every 2 years. I'm not going to go into the merits of the Nobel Prize because I think that Keith Porter should have won the Nobel Prize. With Claude and Palade, the citation was for their discoveries concerning the structure and functional organization of the cell. Doesn't say anything about mammalian cell. Porter went to plants. The person who got it with them was a good scientist named Deduve, who discovered 10 years after the beginning of cell biology, lysosome. And the whole business of autophagia came from that. So there's no doubt that Deduve should have had merited the Nobel Prize. But they have this fixation that you could only give it to 3. So they put Deduve in and took Porter out. I think that was a disaster. Now, there's a very curious thing on internet which called 10 Nobel snubs. The other 10 are physicists. So I took 5 names that we could recognize. Oswald Avery, and McLeod and McCarthy at Rockefeller demonstrated that the transforming principle of pneumococcus was DNA. Didn't get the Nobel Prize. Rosalind Franklin was a crystallographer who showed that DNA was a double-stranded helix. Who hadn't published it, Watson and Crick picked it up. They were in Cambridge or Oxford, and used that in their theory of base pairing, which again deserves the Nobel Prize, but they forgot about her. With Watson and Crick, a X-ray crystallographer got the Nobel Prize, because that was very fashionable. Keith Porter. Dmitry Mendeleev was a Russian chemist who invented the periodic table. My God. When he did that, they were giving Nobel Prizes, but he didn't get it. And finally, Joshua Gibbs is the man who invented thermodynamics, or started thermodynamics. It's all based on his work. So these are 5 people who should have gotten the Nobel Prize and didn't. Now I'm going to show quickly the early, most of them are early pictures of the guinea pig pancreatic cell and identify the players in the game. This is the endoplasmic reticulum. It's a membrane system which has particles on the membrane, outer membranes. It's a complicated system. And the next slide, next slide. Oh, shows it. So, this is the inside of the endoplasmic reticulum. There are 2 membranes, and on the outer surface are these ribosomes. Okay? Between the membranes is the free cytoplasm. So we have membrane limited space, which is the inside, and the cytosol. The cell is not a big balloon with pieces floating inside. Now, the endoplasmic reticulum is a very big constituent of the cell, and the other, these are higher magnification. You see the particles on the outside of the membrane. So this is the space, and this is the cytoplasm, where there are both free ribosomes and other things, cytosol. These are microsomes. They are formed during cell disruption, and they are membrane-limited vacuole tubes, again, seen in cross-section, and many of them have dark contents. This is the bottom of the cell is endoplasmic reticulum, and the top is a collection of membranes without particles. This is called the Golgi. And the Golgi has continuity with the endoplasmic reticulum. This is a transition element. We're losing the granules and will be a vacuole without granule. So the endoplasmic reticulum and the Golgi are membrane systems, vast membrane systems that are interconnected. This is the end of a granule, and the big, this is the cisternae of the Golgi, these flat things, and these are collecting vacuoles. They will fill with secretory protein. And this is the butt of a granule, of another collecting granule, vacuole, which is going to be mature to be a zymogen granule. Now, a lot of biochemistry is done in the Golgi. This is the pancreas. This is the collecting vacuole. And these are different cells. The top of the cell, the apical part, is full of these granules, which really are round. This is an early picture with an artifact of cutting. You see they're oblong. Section was cut in this direction. So you have the granules at the top of the cell surrounding a lumen. Here's a granule emptying into the lumen. First, we see the membrane of the granule up against the lumen cell. So the granule moves through the lumen, their membranes fuse, the granule opens, and the content goes into the cell. So, same as we have migration of granules, and secretion is exocytic. You maintain the integrity of the cell. Milk is all full of, mammary secretion is all full of cell organelles because the cell is disrupted. So this probably was necessary because the pancreas is full of digestive enzymes. And if you have pancreatitis, you're digesting your own pancreas. We're going to get to what the secretory products are later. The granules are actually darker. These dark things are mature zymatic granules. And you see they are simply red membrane-limited vacuoles, which fuse with the membrane and discharge. Okay?